is probably interacting with GLE3 here to evoke this anterior defects. So now, the hypothesis was that they played a role together in anterior morphogenesis. And to test that, we had a few questions. Do MSX and GLE3 interact in the first place? And what is this level of interaction? Now, do they instigate common partners in these functions? And also their role in anterior limb morphogenesis as a whole. Now, for one of these first experiments that we did was to look at this morphological cues at the mutants. So in the wild type, as you naturally expect, you have 5 digits, and you do not have the anterior expression of HOXD12. In the GLE3 heterozygous mutant, you have an slight anterior polydactyly phenotype that is observed, and no anterior expression of HOXD12 as well. When you look at MSX1 mutants, you have no polydactylous phenotype, and no anterior expression of HOXD12. But what is very interesting is that now when you combine this mutation of GLE3 and MSX, you then start to see a polydactylous phenotype. Strong polydactylous phenotype in the combined mutation of MSX1 and GLE3. And there is a significant anterior ectopic expression as well of HOXD12. Now, this indicates thus that MSX1 and GLE3 show synergy in their anterior limb growth in itself. But this is the case of MSX1 and GLE3. So what about MSX2? When you look at MSX1 and MSX2 mutants, you have a limited anterior polydactyly, and there is no anterior expression of HOXD12 as well. In combined mutations of MSX1, 2, and GLE3, now you have this striking and very strong polydactylous phenotype that you did not see in the MSX1 and GLE3 mutants. So you have a much stronger polydactylous phenotype and also a significant anterior ectopic expression of HOXD12. This indicates that MSX1, 2, and GLE3, these genes interact at controlling anterior growth and also the digit number. But also the fact that we had HOXD12 expression ectopically is diagnostic of sonic hedgehog signaling. So one of these things we wanted to look at is how is sonic hedgehog expression being regulated in these mutants? So we did certain analysis, and in the controls, as I had mentioned, sonic hedgehog is only expressed posteriorly and it's not expressed anteriorly. So you have no sonic hedgehog expression in the anterior part of the limb. In the mutants of MSX1 and 2, you have a slight anterior ectopic expression of sonic hedgehog. And this is not present in the heterozygous mutants of GLE3 either. However, in combined mutations of MSX1, 2, and 3, you see a very nice enhanced ectopic expression of sonic hedgehog. So what this really means is that MSX and GLE, they repress a possible anterior sonic hedgehog signaling center from happening. So they are acting together in the repression of this sonic hedgehog signaling anteriorly. But also, the fact that sonic hedgehog is expressed anteriorly in these mutants leads us to think that, is it because of this anterior sonic hedgehog signaling that we are actually having these extra digits? And is it not because of some mesodermal overgrowth? That's because of MSX and GLE3. So is it sonic hedgehog then which is actually responsible for these extra digits that we're seeing in these mutants? So to look at that, we did some analysis independent of sonic hedgehog. So this is the wild type where you have 5 digits again. And in the sonic hedgehog null mutant, you only form the thumb, or digit 1. And as I had mentioned, sonic hedgehog is not required for the development of the thumb in itself. So you only form the thumb. And in mutants where you have reduction of MSX levels, you basically do not rescue the sonic hedgehog null deleterious phenotype. However, when you have a GLE3 heterozygous mutant, you rescue partially the sonic hedgehog null mutant deleterious phenotype, and it's even more striking with reduced alleles of MSX as well, so you increase it even more. But with the quadrupled mutants of MSX1 to GLE3 and sonic hedgehog, you not only rescue it, you almost start to form almost a polydactylous phenotype with 6 to 7 digits. So what this really indicates is that although it's true MSX and GLE3 are showing this synergy, it's independent of the expression of sonic hedgehog as well. So we're able to show that there is a genetic level interaction between MSX and GLE, and this synergy is actually independent of sonic hedgehog as well. So this then leads to further questions naturally. But what level does this gene interaction actually happen? Now, do they form a protein complex by which they're regulating genetic expression? And so we started to look at genetic protein level expressions by looking at protein interactions by PLA and co-IP. One of these experiments we wanted to look at was PLA. But the fact that we didn't have good alleles for MSX and GLE3, we had to resort to tagged alleles of MSX1 and GLE3. And so we were using MSX1 HA allele and ROSA GLE3R. One of the main points I want to make here is that this GLE3R allele is basically ubiquitously expressed throughout the limb, and the MSX1 HA is only expressed points where MSX1 is sexually expressed. So with this technique, we then used PLA. Now PLA, which I will describe here in a nutshell, is basically it allows two proteins in close proximity to be analyzed via fluorescent dots.